Hi everyone, today we are going to talk about allo recognition. Let us try to understand the basic terminology. What is allograft? Allograft, the term is used when a foreign organ such as kidney is transplanted into a non-identical individual of the same species. The transplanted organ is called an allograft. The immune response from the recipient to the allograft is termed as alloimmune response, which is initiated by T cell recognition of alloantigens, commonly known as allorecognition. Allorecognition is the first step of a series of complex events that leads to T cell activation, antibody production, and allograft rejection. Allorecognition can occur by one of the three pathways, that is direct, indirect, and semi-direct. In the direct pathway, recipient's T cells recognize intact allogenic HLAs expressed by the donor cells. In the indirect pathway, T cells recognize peptides derived from donor HLAs presented by recipient antigen presenting cells. In the semi-direct pathway, recipient dendritic cells or other antigen presenting cells acquire intact HLAs from donor cells and present them to recipient T cells. The direct and indirect pathways are well understood in organ transplantation, whereas the semi-direct pathway is not of clinical importance. The direct pathway is very important in the immediate post-transplant period. Without appropriate immunosuppression, a strong and effective allo response would follow, which is primarily due to the high number of recipient T cells that will recognize the graft antigens and causes acute cellular rejection. The indirect pathway of allorecognition may also participate in acute rejection, but it is usually predominant in the late onset of rejection and especially chronic rejection. As long as the allograft is present in the host, the recipient antigen presenting cells can pick up the alloantigen shed from the graft and start alloimmune response. Therefore, maintenance immunosuppression is required for the lifetime of the allograft to prevent late rejection and chronic rejection. So, to summarize, the alloimmune response is initiated by T cell recognition of alloantigens through direct or indirect pathways. Sensitive crossmatch is routinely advised before kidney transplant to detect any significant donor-specific antibodies so that hyperacute rejection can be eliminated. Solid phase-based Luminex assay can further characterize HLA antibodies before and after renal transplant to guide the clinician. In addition to the traditional anti-HLA antibodies, alloreactive and autoreactive antibodies against non-HLA antigens have now been increasingly recognized to play an important role in humoral rejection of allograft. Thank you.